looks like there's a vaccine out that's supposed to protect me against COVID-19. But I hate needles, and I don't know what the COVID-19 vaccine is going to do to my body. Oh, I know. I'm going to do my own research first.
Okay, let's see. Should I get the COVID vaccine ? Oh yeah, something like that. I guess I'll just click the first link that pops up.
No way. The COVID-19 vaccine alters your DNA, funded by the Bob Doyle Research Fund. Vaccine uses microchip technology for surveillance to make you magnetic?
Hey dude, are you okay? You look kinda stressed.
Does the COVID-19 vaccine make you magnetic?
Magnetic? What? No, of course it doesn't. Bro, don't tell me you've been reading the fake news on the internet. You know what you should do? Consult an expert. Actually, my brother's friend's cousin's best friend's sister works as an immunologist. Maybe you can talk to her?
Oh yeah, that sounds like a good idea.
A few moments later.
Hi, Dr. Clemenza. Thank you for meeting with me today. Are all those scary things about the COVID-19 vaccine true?
No problem, Britney. There's definitely a lot of misleading information out there about the COVID vaccine, and it definitely does not cause any of those things that you had mentioned earlier.
Yeah, I'm really confused. Could you tell me what actually happens to my body when I get the vaccine? How does it work?
Yeah, for sure.
Unlike conventional vaccines, which contain attenuated or inactivated pathogens, mRNA vaccines instead use laboratory-synthesized mRNAs. These mRNA molecules are encapsulated in lipid nanoparticles such that, when the vaccine is injected into the deltoid muscle, the mRNA is able to pass through the hydrophobic lipid bilayer and enter the muscle cells. Our own cellular translational machinery will produce the spike protein that the artificial mRNA encodes for. This spike protein is unique to the SARS-CoV-2 virus, but harmless when expressed on its own. The spike protein is then ubiquitinated and degraded by cytosolic proteasomes. The resulting peptide fragments are shuttled from the cytosol to the lumen of the endoplasmic reticulum by TAP1 and TAP2 transport proteins associated with antigen processing. Within the ER lumen, partially folded MHC class I proteins will be waiting as a part of the peptide loading complex. This complex includes several chaperone proteins such as calreticulin, ERP57, epicin, and ERAP, which attaches the protein complex to the TAP transporter. Using TAP-induced pores, spike protein fragments will enter the ER lumen and attempt to bind the peptide binding grooves of MHC1 molecules. A cycle of temporary binding and dissociation will occur until a specific peptide is able to bind with high affinity. This binding triggers the dissociation of the peptide loading complex, and the peptide MHC1 complex will be exported to the cell surface. This is the antigen presentation process that allows specific CD8 T cells to recognize SARS-CoV-2 and mount a primary adaptive immune response. This establishes immunological memory against this particular pathogen, so that you will be protected in the case that you're infected by the real virus.
Wow, that's so cool. This mRNA vaccine stuff sounds like it's really useful in priming the body to fight off pathogens. Is this how my flu shot works as well?
Actually, there are a couple of different types of vaccines.
Instead of mRNA, many vaccines contain attenuated or inactivated viruses. These are taken up by antigen-presenting cells, such as macrophages or dendritic cells, via endocytosis. This forms a vesicle containing the virus. Endosomes will fuse with this vesicle and lower its pH. This activates endosomal proteases that degrade the virus into peptide fragments. At the same time, MHC class II proteins are synthesized in the ER, where they associate with the invariant chain. The invariant chain stabilizes the MHC protein and targets it to low pH endosomes, where it is then cleaved, leaving a small clip peptide that remains bound to the MHC2 molecule, which helps block miscellaneous peptide binding. Once the MHC class II compartment fuses with an endosome containing the viral peptide fragments, a protein known as HLA-DM binds to the MHC2 molecule, allowing the release of the clip peptide and the binding of the viral peptides. The peptide MHC2 complex then localizes to the cell membrane, where the foreign antigen is ready to be presented to specific CD4 T cells that will similarly induce an adaptive immune response.
Hold on. There are two of these MHC molecule thingies? What is this magical health churro? Actually, MHC stands for Major Histocompatibility Complex, and it was named after its function in graft rejection.
The MHC molecule is also known as the human leukocyte antigen, or HLA for short. It binds to foreign peptides, and together this complex is recognized by the T cell receptor. There are two classes of MHC molecules, class I and class II. Both consist of heterodimers and are membrane-bound. Structurally, both are similar to antibodies. The alpha 3 and beta 2M subunits in class 1 and the alpha 2 and beta 2 subunit in class 2 resemble immunoglobulin constant domains. Class 1 has an alpha 1 and an alpha 2 subunit that forms the cleft where peptides bind, similar to alpha 1 and beta 1 subunits in class 2. However, class 1 also has a beta 2 microglobulin subunit that associates non-covalently, whereas class 2 does not. The alpha chain spans the membrane in class 1 in contrast to both alpha and beta chains in class 2. There are clusters of conserved residues in MHC class 1 to stabilize peptide binding. Additionally, there are also anchor residues in both classes to further stabilize the peptide. Class 1 is able to bind to 8 to 10 amino acids, while class 2 is more flexible, binding up to 17 amino acids. In humans, the genes that encode for MHC molecules are found on chromosome 6. For class 1, these genes are HLA-A, HLA-B, and HLA-C. All three encode for different alpha chains that combine with beta-2M subunits to form class 1 MHC molecules. On the other hand, class 2 also has 3 different sets of genes, HLA-DR, HLA-DQ, and HLA-DP. HLA-DR has 2 beta genes, and as a result, there are 4 different types of MHC class 2 molecules. Both MHC class 1 and MHC class 2 genes are polymorphic. There can be hundreds of different alleles for each gene locus. Most people are heterozygous, with co-dominant expression of MHC molecules. The combination of alleles is known as one's MHC haplotype. When transplanting organs, graft rejection may result when there is a mismatch of MHC alleles because the host recognizes the MHC molecules to be foreign antigens and attacks them.
Thank you, Dr. Clemenza. I, Brittany Feng, am so excited to get vaxxed.